Okay, I want to welcome you to the 13th session. We will be following up with it. The 14th will be kind of a summary, an overview of other things and some additional information. We thank you for the people who have attended these classes, and we hope that the information has been useful and will be useful to you. We're going to talk about something that perhaps is not as well talked about in creation. That is the idea of how we determine what the original kinds of animals were. We're going to talk about classification and baraminology, but I've wanted to do this introduction for a long time, southern introduction, because it needs to be said. It needs to be said to set the record straight on what is and not empirical science. Empirical science is validated by repetition of experiments, and the results of those experiments may cause the verification, refutation, or revision of the methods, materials, and conclusions of any scientific finding. Something that is not empirical and cannot be validated and cannot be falsified or confirmed experimentally is not science. It may be something else, it may be art, it may be music, it may be history, but it is certainly not science. There are fundamental assumptions that we make about science. And these fundamental assumptions are pretty straightforward, but I think I've had people argue with me about these assumptions, and I say to them, well, you're not living in the real world, because these are the basic assumptions you make to live in the real world. The ability to have self-consciousness of our real existence. If you don't know whether this is real or not, you either need a home or medication. But at any rate, the ability to have self-consciousness of your real existence, that's a basic premise that underlies science. Either it's real or it's not real. These are a priori assumptions before you ever get started. The external natural world is real. That's something else some people don't believe today. Is real and understandable, at least in some extent. It's not veiled in a mystical cloud of Eastern philosophy. It is for real. Our natural senses, here's another one that is constantly denied today. Our natural senses give us a reasonable and reliable perception of the external world. Without those three basic things there is no way to do science, because there's no way to communicate and there's no way to verify anything. Also, the natural world is orderly and reproducible, and therefore subject to and worthy of systematic investigation. If some days there was gravity and some days there wasn't, some days there was air and some days there wasn't, some days there was this and that, if the laws were suspended every other day, then it wouldn't be reproducible and it wouldn't be worthy of systematic investigation, because no investigation would ever predict what you should wear the next day or whether you should even get up the next day. And the other thing, the laws of logic are valid. There has to be a logical basis for what you're doing in science. It cannot be illogical or irrational or strictly emotional. And observable effects underlie causes, and hence the principle of causality governs the world. And what that means is that cause has its roots in the idea that if you take an action in the world around you, you can make a change. If you want to make a cup of coffee, you can actually go get the water and make the coffee. It'll actually happen. It will occur. When we act, we assume that we can make things happen. And this assumption presupposes that there are regularities. If some days you poured the coffee in and out came wine, that might be amazing, but it would not be very pleasing in the morning maybe. Or eggs. There are regularities in nature. That's how we develop laws and observations. Without these basic principles, it's pretty much a crapshoot. Eastern philosophy has no answer. I mean, their philosophical basis, that's why they're so far behind in science. Okay, back on more. All right, let's talk about the logical limits of science. And science in all type of rational reasoning. Induction. Induction thinking, the limitation of induction we know about. We're talking about induction. What is it? That's using particulars, events of experiments, results, observations, and making a generalization from that using logic. The limitations of induction. Induction is the method science uses. Take the particulars and move to the general. The inductive generalizations cannot be proved to be immune from revision. That is the basic hallmark of science. There is nothing, you know, when the president introduced his program, he put people up there with white coats on so everybody would be impressed and listen to what they said. Unfortunately, these white coats people are not priests. They have no knowledge of an eternal standpoint because they're speaking about science. And science is always open for revision. More experience leads to the modification of scientific generalizations. That is why when someone said scientific studies show on Channel 4, Channel 2, take it with a big grain of salt. Induction cannot be shown to be an error, and inductive inferences cannot be shown to lead to conclusions which are certainly true, even though the premises are true. All right, how about this one? Every day last week, I precisely left for work in the car at 8 a.m. and arrived on time. Therefore, every day I leave for work in my car at 8 o'clock, I will arrive on time. Is that a prediction that you can hang your hat on? Why? There's a lot of missing information that's not in there. Are you taking the parkway? Are you taking the back roads? Are you going to be having a flat tire? You know, the premises of these statements may be logically valid. Yes, it did. It worked every day last week. Yes, I left at 8 and got there on time. But it doesn't mean in this situation because of certain external other variables that may enter in. Is it valid to assume a general principle is correct just because all your data agree with it? Yes or no? No. That's an important thing. Very important for scientists to understand and keep them humble. I do these research projects and then I say, this looks like a very good product. This looks like a good medication to control Ebola or whatever it is. And they put it out into the real world and they start doing clinical studies and it's a flop. Deduction is a classical type of reasoning in that we start from the top down. We take a generalization and work our way down to a series of generalizations and then observations and predictions. However, deductive reasoning can never tell us more than is contained in our assumptions that we start with. And since we devise those assumptions or accept them, deductive inference can only reveal the nature of our constructed systems. So really, deductive reasoning is not scientific methodology, although it is used many times in the conclusions and interpretation of papers. Yes, sir. I'm saying that you can't know science as a truth because science is an ever-evolving device. It's ever-evolving study that involves constantly changing information and parameters. Okay? What I learned when I got out of graduate school at the University of Pittsburgh has been refuted in many areas. It is not the same. The truth I learned in 1976 is now not true. Do you understand? We're not talking about eternal things. This is about strictly scientific things. This is why I'm trying to build a case to show you how impossible it is to make certain predictions from science. Okay. Let's go on. How about this one? Every day I leave for work in my car at 8 a.m. It takes me 45 minutes, and every day I arrive on time. Therefore, every day I leave for work in my car at 8 a.m., I arrive on time. Is the conclusion true or not true? It may not be true. It may not be true. Is it valid to assume this prediction will be true every time? No. Get your mind around that. Our country worships science, and it's not worthy of worship. Our country venerates scientists. They're not worthy of veneration. They're not infallible. The very nature of their study and the very nature of what happens makes them not in the role of that. A famous Canadian-French philosopher, Jacques Barzon, he said many years ago, I read it while I was in the Army, he said, we don't have high priests anymore. We have scientists in white lab coats making statements from Mount Sinai. All right, here's some constraints that will apply to empirical science. Let me flip this over a little bit. Science is limited to what can be observed with the five senses. It cannot be corroborated by observation. If it cannot be corroborated by observation, it's not science. Not empirical science. It may be philosophy. It may be a whole bunch of other things, but it's certainly not empirical science. Science is always limited to the present. Observations and experimentation can only be done in the present. It's the only place your five senses work. Science is limiting us to how a process works, not why. It focuses on mechanisms. This is from a paper from a Greek guy called Apostolos Francos. Very excellent paper at the 1990 ICC. Science is limited. It cannot deal with unique or singular events. Reproducibility and observation are the hallmarks of empirical science. It's like the famous cold fusion. If you remember a few years ago, a guy came out and said, I found cold fusion. Nobody else could reproduce it. So obviously, he didn't find cold fusion. He had a mistake in the calculations or in some of the methodology. It cannot deal with singular events. So when you talk about creation, there's no science to apply to what God did on day one. There's no science to apply because it's God creating ex nihilo, or as Bob likes to say, ex deo, out of himself. Science is always limited by its measurements. Accuracy, reliability, error ranges, standardization, methodological variation. So the accuracy of any conclusion is dependent on its methods, and its methods are always changing. That is the other thing to put in your box. Methods are always changing. Let's see what I got. Science cannot be used to reconstruct history. I don't care what the Science Channel and History Channel says. It can't be. All it can do is take inferences. I'll give you an example. There were many years ago when I worked for the coroner of Allegheny County, there was a guy out haunting the neighborhoods of Pittsburgh called the East End Rapist. I don't know how many, 6, 7, 8, or whatever number of them. I forget all the details. But he was arrested, and as soon as he was arrested, the rape stopped. They put him in jail. Years later, DNA came in. Guess what? Yes, that's right. He's not guilty. You see what I mean by science? Can reconstructing history? Did history change? Did the facts change? No. We just got a new method that helped us to be able to interpret those in a way that was more realistically probably relevant to what actually happened. So all historical evidence is forensic in nature. My boss was Dr. Cyril Weck. And as a forensic scientist, the whodunit never ends until all the evidence is evaluated. So I talked about history is different than science. We can use scientific methods to try to find history. We can go out and take fingerprints and do blood types and do all kinds of things to reconstruct a historical event. But all those limitations that I mentioned to you will make our conclusions to a greater or lesser degree tentative, based on the new piece of evidence that comes along, the new method, the new technique. What does this mean for creation scientists? This is not in his paper, this is my saying. Hold on to your biblical God-given description of events tenaciously. Hold on to your origin models tentatively. The Word of God will not change. The Word of God will not be refuted. The Word of God will never be changed. Science, that's a whole different ball of wax. In fact, I have a comment from Apostle here in his paper. He said, "The game of science never ends. All conclusions are tentative. No matter how much information scientists have, they can never know for certain that their conclusions, since what we know about the real world is always known by the particulars through the inductive process, and nothing is ever known for certain about the real world. Because it's always changing." So when scientists get on television and tell you you're too stupid to decide about global warming, just laugh at them. You're not too stupid. They're just too stupid to explain it. If good scientists should be able to explain the facts. And you can come to the conclusion that perhaps it's just not so, whatever the topic is. All right, well, let's talk about classification of biological organisms. I gave you all that preface because it has everything to do with what evolutionists and creationists do with evidence they find in the real world. Historical evidence. Why do we do classification? Is it just for fun? Well, no, because one of the things we gain from it is we can decide whether certain organisms are related to each other. We can decide whether a particular organism will, a particular animal or specimen will thrive in a certain environment or not. And there have been about 1.5 million organisms have been discovered, and there's at least 10 to 100 million kinds of those in the earth today. That's just a guesstimate. A guesstimate. All right, let's talk about the history. You know who the first scientist to classify organisms was? Adam. Genesis 2:18. And the Lord God said, it is not good for that man, the man should alone. I will make him a helpmeet for him. And out of the ground the Lord God formed every beast of the field and every fowl of the air and brought them unto Adam to see what he would call them. And whatsoever Adam called them, called the living creature, that was the name thereof. He was the first classification biologist. But for Adam, after he gave names to all the cattle and the fowl of the air and to every beast of the field, but for Adam there was not found a helpmeet for him. No suitable biological reproductive pair could be found for Adam. I think God did this because he wanted Adam to see that he was different than the other things he created. Personally, it's a personal opinion, it's not. All right, well, let's talk about the classification. It was called a natural classification. What that means is that it was based on names given by an individual to the living things that they saw within their environment, personal environment. It's notable what Adam named, but it's also interesting what he did not name. He did not name plants, sea creatures, insects, etc. He only named those items, the creatures of the field, the fowls of the air, that were directly in his environment. There was a personal type of classification. So classification should be said to put things into the same groups. The science of classification is called taxonomy. That's naming and grouping organisms. The first guy to do this in a formalized method was Aristotle in 4000 BC. And he created two kingdoms, the animal kingdom and the plant kingdom. The animal kingdom was anything that lived on the land, in the water, and the air. The plant kingdom was based on the shape of their stems. That's his classification system. This was the first written formal classification system. This is his classification system. You can see with the laser pointer, life divided to kingdom Animalia, kingdom of Plantis, and then where they live, what they eat. Herbivoria, what kind of animal is that? Plant eater, right. Carnivoria, meat eater. What's an omnivora? Eats whatever. Eats everything. Whereas the kingdom of plants were a xerophyte, which mean dry environments, a hygrophyte, which would mean moist environments, and a hydrophyte, which mean water, nearly 100% water. And you can see the divisions of trees, bushes, and herbs. That was the original first classification done by Aristotle in 4000 BC. And I can say, that's pretty good. That's not bad for just gross observation. Observation moved ahead, though, with the classification brought out by Carolinus Linnaeus in 1700s. He lived 1707 and 1778, and his classification system established a system of classifying and naming organisms based on their structural similarities of the organisms. He also developed the idea of the binomial nomenclature. Binomial nomenclature is a big word, there's two names. It's the genus and species of the name of the particular animal we're talking about or a particular plant. So that system was developed by Linnaeus. He had two kingdoms in his system, one regime vegetal and regime animal. Here's a picture of his animal classification. I'm sorry, this is the actual published classification he put out in 1700. He had 2.5 million kinds in that. Modern classifications are not based on the Linnaean system. Modern classifications are based on a Darwinian principle of descent. Charles Darwin was not the father of this approach. Others preceded him in setting up the concept that things proceed from simple to complex. His grandfather, Erasmus Darwin, 1732-1802, produced a book called Zoonomia in 1796. And that's where he explored the idea of transmutation of species. Transmutation, one thing changing to another. Okay? Then Jean-Baptiste Lamarck in 1744-1729 produced a book called Philosophical Zoology, 1809. He saw all life, past and present, in the form of a hierarchy, from the lowest organisms with the least specialization to the highest with the greatest specialization. He called this the great chain of being. So Darwin, although he was first in developing certain things, really there were principles set down. His own grandfather set down some of these principles. All right, this is a descent spindle diagram for some of the vertebrates. And what you can see is, let me take this back. You can see that it starts back here in the at the Cambrian in the unmisty past, and the differentiations go off. What you see here is probably the actual organisms, but what you see here is the hypothetical differentiations of each of those progenitors. There'd be a progenitor of this group, progenitor at this thing, at this one, and the thickness indicates the amount of extant species at the time. Classification systems have never been static. For most of the 20th and the 21st century, taxonomists have been attempting to correlate descent from a common ancestor to all living things. They have not been successful in that task. Thus, for those of you who don't know, there are two systems really at work. One system is using binomial nomenclature, and there's an official international book listing those classifications, and its purpose is to have a dictionary. So when scientists talk about a specific genus of species, everyone's talking about the same thing. Okay. So it's a naming system. The other one tries to link all organisms by various characteristics to a great chain of evolutionary relationships, both extinct and living. That is the evolutionary approach of simple to complex. One is a naming system, one is the other. These are the classification systems that, oops. The wrong button there. Oh, I keep hitting the wrong button. Come on, Dennis. Hey. My pointer's not working. Oh, can you? All right, well, we'll go back then. This is a Linnaeus classification. This is Haeckel, a famous evolutionary scientist. These are some of the other guys and their dates, their kingdoms. In reviewing what's in the teaching literature, I would say most of what's being taught in high school and maybe some colleges is the 5 kingdom system. That's what I found the most references on. But actually the one being used by scientists was 3 domain system. And you can see the difference between how they flip back and forth. Haeckel had protists, Chatton had prokarya, Copeland Monera, Woese has now divided into bacteria and archaea. So these systems have varied as to their determination, especially their usefulness or non-usefulness in evolutionary lineage. Comparing the two systems, one can see that the 3 domain system is the currently at lie. There's a domain of bacteria, archaea, the ancient organisms, and then all the way back here, universal ancestor. He doesn't have a name, he's just there. How do we know there's a universal ancestor? Because that's the product of the a priori assumption, which says that all things came from a universal ancestor. So if you had an universal ancestor, and that's the truth, there has to be a universal ancestor. It's logical, right? And now you see the different domains, the fungi, the animals, the plants, all different groups in there. A lot of this stuff, go ahead, sir. Right here. No, they in this particular chart, they didn't want to, they didn't bother to put in some of the ancient forms. And same with the domain bacteria, which, you know, obviously is a huge, huge, huge domain. All right, as far as what you probably learned in school, most of us learned, well, this will show you if I can get out this, back over here. Life, domain, and then most of us learned the kingdom, phylum, class, order, family, genus, and species. There are two good acronyms you can use for this. One is King Philip came over for good spaghetti, or kids playing catch on the freeway get smashed. Depending on your particular mindset, you might want to choose one of the others and surprise your children or grandchildren the fact that you know that kingdom, phylum, class, order, family, genus, and species. Because you can remember King Philip came over for good spaghetti, or kids playing catch on the freeway get smashed. And there you go. That's your tip to impress the kids. But at any rate, most of us, and then of course the name of the animals, the genus, and species. Here's an example using this system. Man is in the kingdom Animalia. Box elder tree is in the kingdom Plantae. Bobcats in Animalia. A Canadian lynx is Animalia. These are chordata, which are the ones with a single spinal cord, and you can see all have chordata, chordata, chordata. And that's very interesting because when I started biology in undergraduate school in 69, we had an animal called Mogulum manhattanensis, or the sea squirt. Mogulum manhattanensis is very interesting in that it only has a notochord or a member of the chordata during its juvenile life. It becomes sessile and feeds off passing things during the rest of its life. So it was classified as a chordata, even though for most of its adult life it was not a chordata. The actual nervous system went away when it went to the adult. Very interesting, the central nervous system. But anyway, you can see as you come down these classifications that we got, it would be Homo sapiens. And you can see if you have two in the same group, the cat Felidae, lynx and lynx. Look at that. The bobcat, that means what? They're the same what? Genus. But one is Rufus, the other is Cometanensis. So it hopes to see how they use it in the naming mechanisms. Modern methods of trying to tie together primitive or extinct and living organisms, a method that's used as a metric is called cladistics. Cladistics is not interested in a lot of things except common ancestry. And you read there, it is an approach to classification which organisms are grouped together based on whether or not they have one or more shared unique characteristics. That's called a character state. And usually that's a plus or minus. Character state is something that the biologist determines these animals have or don't have to be a member of that group. At any rate, characteristics that come from the group's last common ancestor. Has nothing to do with the relationship. The whole assumption of cladistics is based on our cladistical modeling. What was the last common ancestor? That are not present in more distant ancestors. So if that trait is carried through, then that can't possibly be the more primitive form of the organism. So therefore, members of a group are thought to share a common history. These are represented by cladograms. These are two examples of vertical cladograms. Again, at the bottom is the primitive or unnamed organism. These are the splitting parts. You can see that in this one, if you go up, there is a common ancestor for A and B, who is not the common ancestor for C. But there is a common ancestor for A, B, and C. You see it probably better in this representation here. A cladogram is not an evolutionary tree. This is not me talking. This is the references. Because it does not show how ancestors are related to their descendants or how much they have changed. Many evolutionary trees can be inferred from a single cladogram. Say that again. Many evolutionary trees can be inferred from the same cladogram. Because of the astronomical number of possible cladograms, there are all algorithms, which are numerical calculations processes done on a computer through a program of some kind. Algorithms cannot guarantee that the solution is the overall best solution. Again, these are quotes, a non-optimal cladogram will be selected if the program settles on a local minimum rather than the desired global minimum. And what that simply means is it may decide to fudge it one way or the other, whichever looks better, as far as what the person wants to do with their particular program. But I want you to understand, you can have lots of trees in the same cladogram. But even so, trying to select which cladogram is right is a big problem. So they have to use what they call an annealing program. Annealing means, putting it together. And that improves to increase the likelihood that the selected cladogram is the optimal one. This brings up something I do not want to discuss. It's under your next notes on page 13, parsimony and evolutionary trees. We're not going to get into that. You can read that at your pleasure. But evolutionists strive to have the fewest number of steps as possible in the evolutionary tree because they feel it takes so much more information diversification if you have more steps. That's why they're talking about a global minimum versus the local minimum versus a global minimum. That's talking about parsimony. And they have to use a annealing process. So how comfortable do you feel about the description of the historic reality of these relationships? Here's an example of the problem. This is from the literature. This is not mine again. Cladistics problem example. And there you go. We have two. The first cladogram used to be the generally accepted for relations among turtles, lizards, crocodilians, and birds. Started at testuines here, and the green says that is the father or the parent of the turtles, until it comes down to the dad says they're parents then for all the other ones. So it appears that the hypothesis, if it's correct, the last common ancestor of turtles and birds is right here. That's the last common ancestor. That's based on basic morphology, not. Okay. Down here on the molecular level, you get a completely different cladogram, relationary history. Again, now you have this particular junction being the ancestor for all these groups, all the way down to the birds. So now the birds share a common ancestor. And the quote I liked from the paper was this: if this is accurate, then the last common ancestor of turtles and birds lived later than the last common ancestor of lizards and birds. Since the cladogram provide competing accounts of real events, at most, one of them is correct. I like the way he said that. At most, one of them is correct. Doesn't mean any of them are correct. Finally, again from the literature, cladistics, either generally or in specific applications, have been criticized from its beginnings. Decisions as to whether a particular character states are homologous, a precondition of synchromorphy, which is part of who was the last common ancestor, have been challenged as involving circular reasoning and subjective judgments. Surprise, surprise, surprise. It's history, not science. It's history. We're arguing about history now. Baraminology. What is baraminology? How did it come about? Where did it start? Well, you'll be happy to know that some of the most important papers were presented right here in Pittsburgh at the ICC, 1989, 1990, especially 1990 ICC. It was based on a very basic kind of a priori assumption, and that is that biological organisms were created in their kinds by God in the creation week. Okay? That's when the kinds were set up. And then afterwards, they diversified very, very quickly over a short period of time, interrupted by a catastrophe known as Noah's flood. That is exactly the upside-down opposite from the evolutionary or standard model. I usually don't touch on evolutionary because I use standard model commonly when I'm talking to people because evolution is a standard model. But evolution standard model traces all life back as a single tree to a single primitive life form. So it is dramatically different. What we're looking at, there were original kinds that were different than each other, and that there was variation within the kind, and that variation was affected not only by the environment that they lived in before the flood, it was affected by the flood and the environment they lived in after the flood. Creation scientists also agree that most, if not nearly all, the sedimentary layers, that's the stuff that's laid down by water, belongs to the flood and the immediate post-flood era. The black arrows on the accompanying chart of the standard geological model column indicate the major views of when the boundary occurred. The majority of young Earth creation geologists would probably fall between the 2 hours at the top of the chart. This is from CMI, for those who know what CMI is. And there you see probably the two prevailing views as the flood, post-flood boundary as reflected in the standard naming of the geological strata by the mainstream standard model people. The idea of a baramin, that word baramin, comes from the word bara, which means to create, and min means kind. It was a term that was first created for the created kind, and it was attributed to Frank Marsh. In 1941, he coined this term. Marsh's ideas have begun to flourish over the last 2 decades. There's a very large creationist group in Germany, so you know that we're not alone in the United States, called Wort und Wissen, and it's produced a book of systematic papers called Typen des Lebens, in which they apply Marsh's ideas to groups of plants and animals. So this is a study, baraminism and baraminology is a study that's being conducted not only here, but across the globe among creation scientists. The International Conference on Creation, or ICCs, have seen many primary papers on baraminology. These have included what I consider to be the 2 seminal papers. One, Discontinuity Systematics by Walter Riemann in the second IC in 1990, and that was the basis of the classification system. And the second one was Baraminology, a Young Earth Creation Biosystematic by Dr. Kurt Wise. This paper gave the discipline its name, and that the biblical criteria for classification using Riemann's papers as basis. Now, in order for us to understand how baraminology works, I have to take you on an exercise. You may not like it. Nobody likes exercise. But we need to do it. So we're going to use a paper by Mr. Baraminology, Dr. Charles Todd Wood. He presented in Creation, a magazine. And it's a tutorial he did. And so we're going to talk a little bit about how baraminology works, how creation scientists try to classify things to the original kind. An unambiguous criterion for what a baramin is would make research a lot easier. But even the use of hybridization criterion has serious limitations, even among living organisms. It's not applicable to asexual or fossil organisms. Because of these problems, creation biologists today approximate the limits of a baramin using a suite of characteristics to assist them and employ the 3 terms that were derived from Marsh's baramin word. Those 3 terms are a monobaram. A monobaramin is a group of organisms that share a continuity, either genetic or phonetic. The word phonetic means of or related to a taxonomic analysis that emphasizes the overall similarities among biological taxa without regard to phylogenetic relationships. Phylogenetic relationships, the evolutionary tree. Okay? An apobaram is a group of organisms that's discontinuous with everybody else. So a monobaram, they share all these things they like. an apobarum is a group of organisms that they're separated from all the other organisms by some characteristics. For example, the example they gave in the paper, Dr. Wood gave in the paper, was bats are an example of a group of unrelated mammals that we don't know how many baramins of bats God created, and baramists refer to these bats as an apobaramin. Finally, the holobaramin. The holobaramin is where everybody wants to go, and that, roughly speaking, is the Genesis kind. Technically, it simply combines the definitions of monobaramin and holobaramin, which means it has organisms that share all the continuity among themselves and are completely discontinuous with all other organisms. And that is the original kind. Now, how do you do that? Well, the study he did was he wanted to do living organisms. They're a lot easier to do hybridization on. Dead things tend not to hybridize. Joke, forget it. All right, anyway, Poaceae, or Poaceae, are the class of grasses that he wanted to work with. And the grasses include all the cereal crops like maize, oats, wheat, barley, rye, and sugarcane. There's 10,000 species in the family Poaceae, or Poaceae. Either way is correct to say it. The first thing a baraminologist does is take into account biblical considerations. Again, it's history. So if you're going to deal with history, you got to use a history book. So I use the Bible to start with. It made the identification and interpretation of baramins. You should consult all the various translations, all the sources, including primary sources. In our case of grasses, this is what Dr. Wood found. The words used that are translated grass, grasses, vegetation, barley, rye. And you can see between the various versions of the English Bibles, the KJV, the New King James, the American Standard Version, the Revised Standard, and the New International, you can see the differences in how the same word from the Hebrew in these verses are translated. Between grass and vegetation, corn, it's translated grain, sheaves, translated NIV, and you can see all these things. The important factor is when you start your study, you're starting with the Bible. See if there's information the Bible can give you about what you're going to study. Based on this, Dr. Wood said it appears that it does not directly address the origin of grasses, Poaceae. For if God created all these plants, whether herbaceous, that means herb-like, or woody, trees and things like that, then he most likely created many plants of different baramins at one time. There is no apobaramitic division between them seen here in the Bible. And what was an apobaramin? Was an apobaramin? Oh, you're not going to... Come on, guys. Something that is discontinuous with everything else. All right, here we go. One of the things they do, and again, only with living organisms because you can't hybridize the dead, there are many works in the published scientific literature about hybridization. And to display hybridization information, baraminologists use a graphical tool called a hybridogram. Hybridogram is done on a graph paper or a computer spreadsheet. The species being studied is listed down the left side and across the top. You record successful hybridizations by filling in the appropriate cells. Dr. Wood decided he could not use all 10,000 species, so he used an approximation by choosing the 46 tribes of grass as defined by Watson and Dallwitz in their paper. I guess it's a standardized conglomerate of how to combine the grasses. Hybridogram tribes and grasses. The names of the organisms on the top and bottom of the chart are the same. Black squares represent reports of intertribal hybridizations. Gray squares indicate two tribes are known to hybridize in the same third tribe. And open squares means there's no reported hybrids. Does that mean there are no hybrids? Because there's no reported ones? It's a science. I asked you the question we learned at work. What about science? Because it's not reported, is it not true? It's just not reported yet. You got to think like good critical scientists. I want you to think this way every time you listen to that stuff on the news and take a good critical approach. So here's the hybridogram he came up with. Across the top to bottom, left to right. This is the first one on top. This is the first one going down. This is the same one on this side. So obviously, in the square here, you can see that the same organism should hybridize with itself. That should be an obvious given. If it doesn't, check your methods. At any rate, this is what the chart looks like. And you can see some hybridize with other groups, and some don't hybridize with anybody. Okay. Dr. Wood chose only to take the 12 tribes of the 46 tribes that hybridized. He rationalized that the 12 tribes that did hybridize, among many groups, comprised 7,220 species, so that's about 72% of the family Poaceae. He reasoned that the remaining tribes not related by hybridization are small in number. However, two of those tribes, the Bambuceae, which includes the bamboos, and the Stipiae, which includes the rice grasses, are very large. So we're still dropping out some large category of animals, or plants rather. So he continued with the hypothesis that 70% of the grasses in the 12 tribes that did hybridize form a monobaramin. A monobaramin is things that all share things in common but are different from everybody else. Okay? Finally, one of the metrics they use, like they use cladistics, use a metric. The metric used in baraminology is a thing called baraminic distance. And baraminic distance is a metric that summarizes the data of hybridization, or it can be used to summarize any of the character state between the various taxa. It's a systematic arrangement column. Column represents a particular characteristic, such as two shape or head side. So across the top, you have a characteristic, head, square or round. Underneath, German, square, yes. Non-German, round. No, I'm just joking. They'd be classified that way, down this one side. You understand? And you'd give it a number. Everything has to have a number because you can't analyze it on a computer without a number. So they give the 1 or 2, and we'll talk about that in a second. Example, oat flowers, character column could have a character state of bisexual, while maize flowers may have a character state of 2 as unisexual. Character states are always coded numerically. At this point, Dr. Wood needed to find a systematic database that included a lot of characteristics for the grass. And he found one from a baraminic group called the Grass Phylogeny Working Group, GPWG, we'll call it from now on. The barometric distance is the distance between two species, is the percentage of their characters in which the two species differ in their character states. I'll say that again. The distance is two species, how different are they? What is the percentage of differences in their character states? An example that he gave in his paper, if we look at A and C, he plotted it. You can see that the barometric distance as a plot, A and C are closer together than they are closer to B, F, D, and E. So A and C may be related to each other, but B, F, and D may or may not be related to A and C. That's just for one characteristic. Okay, he's showing you one characteristic. And it's really a statistic that's based on the square of the correlation. And he talks about that. Now, Dr. Wood needed to have some computer software to evaluate that. And he did this program called BDST to evaluate the distances. And he calculated based on character relevance. I know this is hard and it seems boring, but I'm trying to get you to understand just how difficult it probably is to get back to what we're talking about. It ain't as easy as you think. Character relevance simply says those character states, 1 or 2, well, if some of your species don't have those character states, they should not be figured into calculation for a barometric distance. So they came up with what I would call a confidence limit of 95%. If characters that have 95% of them have the same character in the relevance test, great. If they don't, you get rid of that characteristic as a comparative. He eliminated low relevance characteristics, and he eliminated 4,906 characters from the table. 4,906 characters. Finally, he calculated the barometric distance for the remaining characters, and he outputted it to a plain text file, and it can be put into a spreadsheet or other mathematical software for further analysis. Two charts are following. We're almost done here. Hold on with me. One is for molecular and morphological data, and the other is just for morphological data. For both of these charts, what you're going to see are black squares, and then you're going to see or dark gray, black squares. Then you're going to see circles, which mean no negative correlation. They're not correlated to each other. There's no correlation and positive correlation. And up the one side are the names of the organisms. Across the top are the same organisms. Put it against each other to see how they compare. And this is the DNA and morphological. The only thing I got to say about this, my little marker went away again. All right, you can see that there are lots and lots of these. Lots and lots of these. I don't touch the buttons. Lots and lots of these. These are the outgroups right here at the bottom. And if you take this, you take where I'm pointing right here and flip it so that it starts at this side and moves backwards. So the first one here represents the first column. The last one here represents the last column, which is at the bottom. You can see that, as we would expect, they do not have a positive relationship to the rest of the grasses. But unfortunately, they also have a lot of grays in those outliers and that outgroup, which is a control group. And you also can see there are two groups of organisms which have a negative correspondence, but also share some common shared characteristics in as far as a positive relevance. So what did he say about this? Based on this, Dr. Woods said 98% of those in the chart had a positive correlation, 6% had a negative correlation. 53% of the 248 species pairs between grasses and the outgroup also had a positive correlation. Not good. Not good. If you can't separate your control group from your test group, it's probably not going to give you anything to interpret. So he went back. He did not expect such a high correlation in the outgroup. He believed the results were indicative that the non-Poaceae genera included in the data set might be members of a monobarum together with grasses. This would be a surprising result since grasses are considered to form a well-defined group by the standard model. He evaluated the character distances and eliminated the DNA and used morphological shape, what it looks like, characters. And he said, of the 7,000 characters in figure 3A, 6,900 or so came from DNA, and only 53 of the characters came from morphological. And of those 53 characters, 21 had to be eliminated because of low relevance, which only left them with 32 remaining morphological characteristics to compare the groups with. And as you can see, there's a lot of positive correlation. And it also looks like that the outgroups are also showing. But in addition, you can see that there are some problems with correlation with the one group here. If you go back, let me go back here. These two groups here, they have a negative correlation with all the other grasses, but a positive correlation with themselves. I found that nearly every one of the grasses shares significant positive correlation with the other grasses, but significant negative correlation with the outgroup. Two noticeable exceptions are there's two groups remain. We're not going to go into and both of them which have negative correlation with most of the other grasses, but significant positive correlation with the four outgroup genera. From morphological analysis, first of all, Poaceae, excluding those two tribes, form a coherent monobaramin and apobaramin, suggesting that the grass species in this group are members of single holobaramin, the single original kind. Second, negative barometric distance correlation indicates that the tribes that were eliminated are not members of the grass holobarum. The position of two of them are Ferris and Ferry and that cherry borer are unclear. Thirdly, molecular sequence data biases barometric analysis toward too much similarity. His conclusion is DNA sequence. The author says that strongly suggests that researchers should not rely on sequence similarity, DNA similarities, in determining barometric distances. Okay. Okay, let's go into the discussion. And let's try to bring this all home and make sense of it. Okay? What are evolutionists looking to find? You can answer. Let's speak out. What are they trying to do when they're looking at a tree? What are they trying to find? What's the goal? The original what? The universal right answers. What is the creation scientist trying to do? Find the original kind. They share that goal together. It's a historical, retrospective study, looking back on the basis of the historical evidence and what current evidence you have. And as such, they share the same problems. Those problems can be looked in two groups: extinct organisms, that's organisms that are croaked, and extant, those that are living. Empirical research data is limited or non-existent for extinct organisms. For example, what we commonly use in the relating organism: molecular, morphological, biochemistry, behavior, mating, and hybridization, the most common ways to link living organisms to each other. And they're just not existent for extinct organisms. Okay? It's hard to get those trilobites to mate. All such extinct organism data is selective. You've lost all the soft tissue parts. The DNA is gone. And they have to be presumed from currently living examples as being a related organism. All right. All evidence for extinct organisms is inductive and forensic, and thus is methodologically dependent. How about living things, extant things? Morphological problems always have been a problem when classifying things, is that things that look alike are not necessarily alike in their history of their development due to isolation, founder effects, bottlenecks, and environmental pressures. Suppose one of my characteristics was the ability to fly. How many different groups would fall into that? Mammals? Who else? Birds? Who else? Reptiles? Reptiles? Who else? Insects. They all fly, but they all use different methodologies and different ways to do it. That helped to clear it up maybe a little bit. Okay. Also, founder effect and bottlenecks. If you were here in our first lecture, we talked about what that does to genetic components within an organism. You may lose information in a founder effect or a bottleneck effect. Founder effect is due to a catastrophe like Noah's flood. Founder effect is two birds arrive on an island, mom and dad, and they have all the children, but they aren't related to all their other ancestors. Biochemical genetic problems with extant species. They are monstrous, no matter what they tell you. First of all, we are in the infant stages of doing genetic classification. We are really in the infant stages. We have no real good evidence on how much genetic stability based on the kind of methodologies we use over time and what that stability means. We have no way of definitely relating it to relatedness because the methodology is always changing. DNA, if you saw the paper this week, what did they find from the 911 site? What were they able to identify? Did you see the headlines in the front paper? All right, well, I'll bring you up to date. They found, from one of the pieces of tissues from the unidentified dead people was able to be finally identified as a CMU student who died in the tower at 911. Why couldn't they identify it before? They just got some new DNA hybridization techniques and methodologies that could resolve the discrepancies. All right, blind spots. Evolutionary blind spots have been well documented in creation literature when it comes to assumptions about ancestors. What about baraminology blind spots? This is a baraminology tree for cats, saber-tooth and the main cat kind, indicating the, I guess, back here is creation, and it all stayed nice in one little group all the way up to the flood. No diversification, no separation, no isolation. I don't know how you know that, but apparently you do. And when they got to the flood, certain groups died out and certain survived. The main cat kind. Certain certain things were lost. Okay. There's a problem with this chart. And the problem is that here are the problems that are being missed. At creation, everything was how? Perfect. There was no disease, no mutation, no genetic degradation of any kind, no predation, no pathological processes, no birth malformations, no genetic diseases. Get the picture? It does not show the effects of the fall. What did the fall have effect on those organisms? Just this predation alone. And all those parasites that live in us, they call parasites, probably have a beneficial purpose when they were created by God. They're parasites now, but they weren't parasites then. Do you understand where we're going with this? Okay, post-fall, something really big happened because everything that I listed in one no longer existed. So that means they no longer resembled their created kinds in morphology, behavior, biochemistry, genetics. I would dare say that was a big change of which we have no definite measure of its effect on living things. Finally, non-survivors of the flood. What happened to all the animals that croaked in the flood? They all contained genetic information. All of those animals, were the vast majority of the genetic information was destroyed during the flood. Only 2 by 2, or 7 of the clean kind, came on the ark. The representatives of the ark did not reflect or contain the genetic history of all the created kinds that were at day 6. There were 2,000 years from the creation of the flood of generations of sin's effect, isolation, distribution, selection to work on the genetic libraries of those kinds taken on the ark. The Bible does not indicate that God recreated the original kinds to take on the ark. It indicates the kinds as they were when Noah was alive or what he took on the ark. Post-flood. Again, we're not sure what kinds of the ark survived anyway, in their original type. And how many were segregated, isolated, or changed, and genetic information lost is lost for good as far as lineage. Finally, there are some important concluding comments. This does not mean baraminology is worthless. What it means is, like all historical studies, it has to be taken with a grain of salt. There are real limits to biological change. We know you cannot have limitless changes between organisms. We know there are limits to change. The original kinds were different in ways that we cannot be certain for classification purposes. Simple organisms did not transpute to more complex. This is a very critical failing point of evolution. Useful information must be present at the time it is selected for survival. It cannot appear beforehand because there's no foresight and no direction in evolution. You have to have the advantageous mutation available at the time that the gigantic environmental impact happens. That asteroid hits the Earth, the clouds are filled with dust, the dinosaurs didn't make it. It didn't matter how hard they tried, they all died. They all died why? There's no way they could come up with a way to change their processes and the genetic information like that. Environmental changes happen like that. So, okay, onward. Creation is evident in the information and complexity of living things we see in the world around us. Information never comes from non-information. Information that is very complex has a complex source and a complex coding mechanism, and such we see in living things. Fossil organisms are highly developed and information-rich from the beginning based on their morphology. Only degradation can be seen in the number and types present in the fossil record. What is one of the great evidences of the fossil record? Mass what? Extinctions. Mass extinctions dominate the fossil record. Extinction continues in this current observable scientific age, but we do not see accumulating useful information. The Bible was not written as a text on taxonomy or biology. Therefore, great care must be used in using the Scripture for classification purposes. That's from another paper from there, from CMI, on creationist literature. But baraminology can be a useful endeavor in the investigation of relationships of living species. However, its use in the study of historic original kind relations are fraught with difficulties. And that's an understatement. All right, that's it. Question.